This protocol is significant for determining the glycolytic function and mitochondrial respiration in Candida albicans. The main advantage of this technique is that it allows for mitochondrial function to be measured without the need to purify mitochondria from Candida albicans.
This method can be optimized to investigate the effects of genetic manipulation or chemical modulators on mitochondrial and glycolytic pathways in Candida albicans or other fungal pathogens. This technique is relatively simple. However, depending on the organism, the user may have to optimize the cell number and the concentration of mitochondrial inhibitors in their assay.
In a laminar flow hood, dissolve poly-D-lysine in tissue-grade water to a final concentration of 50 micrograms per milliliter. Mix this solution well and aliquot it into 1.5 milliliter microcentrifuge tubes, making sure to aliquot at least 1.3 milliliters per tube. Add 50 microliters of this mixture to each well of the assay plate. Cover the lid and incubate at room temperature for 1 to 2 hours. Then, aspirate the solution. Rinse the wells of the assay plate once with 500 microliters of sterile tissue culture grade water. Open the lid and allow the wells to air dry. If not using the plate the day it is prepared, store it at 4 degrees Celsius for a maximum of 2 to 3 days. The day before the experiment, open